Hi everyone, welcome to Reason with Science. I'm your host Jitendra. This episode is with William Ratcliff. He is an associate professor at the School of Biological Science at Georgia Tech. Will studies the physics that constrains multicellular structures and examines the minute changes in certain yeast cells that allows them to become multicellular. Here we talk about origin of life, emergence of complexity, eukaryogenesis, how cells become multicellular, and major questions regarding how multicellularity evolves. Enjoy the conversation, share and subscribe to support the podcast. Thank you for listening. Hi, Will. Welcome to the podcast.
Thank you so much. It's a pleasure to be here.
So, thinking of this diversity of life on the planet, what is like because of course one theory that we have, or one major theory that in biology that we have, is the evolutionary theory, and according to that theory, what we understand is that all the life forms, they evolved from one common ancestor, right? um, but before we talk about how that one common ancestor can transform in all the diverse life forms, let's kind of talk about like how would that common ancestor would emerge. So the question of origin of life. So what are your views and understanding about the topic?
Yeah. It's a great question. In many ways, I think this is the question of the century, right? It's one of those big questions that the more we work on it, the more we chip away at sort of possibilities. And I think we can learn a lot about how it could potentially occur. But it's one of those things that's very, very difficult to pin down how it actually happened on Earth, because it happened so dang long ago. We know that we had full-blown cells, in some of our earliest fossils, in which you have, seafloor sediments, often around hydrothermal vents 3.5 billion years ago, which contain microfossils that look like cells. And if you look at them, you see they have depleted carbon. So this is depleted carbon is evidence that the carbon went through biological processes and enzymes, which tend to exclude heavier isotopes of carbon. So, it looks like a fossil, and it's got the isotopic signature of fossils. So we're pretty confident that we had, bacteria-like organisms pretty quickly, and likely it happened much earlier than that. By then, it was seemingly life had kind of taken over planet Earth. So it happened fast. And by and large, there's sort of, I think, two different schools of thought on how life arose, right? We have the sort of replication first school of thought, which argues, and I think quite sensibly, that life is Darwinized chemistry, right? You have chemistry which has the potential to open a Darwinian process. A Darwinian process is one in which you have an entity. It doesn't have to be living. This can be anything. This is actually an algorithm, right? It replicates. The offspring of it varies somewhat from one another, right? That variation is at least somewhat heritable, and that variation affects that entity's frequency in the population. And there's really only two fundamental ways to affect your frequency in a population. You can either reproduce more or die less. That's really, it all boils down to that. And so if you have something which replicates, which has heritable variation, which affects fitness is that change in frequency over time, right? Then you have Darwinian evolution. You have populations that get more fit on average over time, and you have the ability to accumulate sequential adaptations, which is kind of how you add information into biological systems. I'll note that, I said it doesn't have to be biological. Clearly, in the case of biology, in the case of life, this is just an unmitigated runaway success story. But this works in other places too, right? It works with information. The whole concept of memes is the idea that, like, information, it has replication, it has variation, it has heredity. Someone that has a really funny idea on the internet can get it spread to millions, and others will modify it, and it will, increase or decrease its rate of spread. That's a Darwinian process. It happens with languages, right? The whole Indo-European language group has only a 10,000-year-old ancestor, and there's a bunch of languages in there. And you can have, you can employ it in computer programs. So this is just, it's kind of just it's almost like an F equals MA. It's just a rule about the way the universe works. Anything which has these criteria can potentially evolve. So we know that, life is essentially Darwinized chemistry. And so one school of thought is that you have replication of essentially simple chemical replicators first. And that's sort of, spiral in a way that different people argue occurred different ways, right? It's all kind of plausibility arguments at this stage. You accumulate adaptations, which results in increasingly sophisticated life. The other sort of school of thought is that you have a metabolism first approach, right? That you basically have an energetic gradient. Some people argue that planet Earth is a giant battery, that you have this iron core, and you basically have electrons flowing from this iron core through essentially through the oceans, through the atmosphere, and escaping into space. And so you just have this gradual flow of energy from the planet at planetary scales, which, if there is any chemistry out there that can take advantage of that gradient, is able to use it to drive chemical processes. And so that metabolism sort of came first. It's a part of the environment, and life kind of arose as a way to dissipate that energetic disequilibrium. I think Nick Lane's ideas on this are super good. I highly recommend listening to his talks. There's a number of other people that have worked on this, that all their work is really cool. To be honest, though, I'm not an expert in the origin of life. I'm just a consumer when it comes to the origin of life. I like to see what other people are working on. I do find a lot of satisfaction in Nick's arguments of having basically hydrothermal vent conditions in which you have essentially small chambers in rock, which are roughly the size of a cell. You have these Fischer-Tropsch processes of just straight up chemistry, in which you have carbon polymers that are created on the surfaces of these rocks, in which you already have minerals, which allow for certain kinds of catalysis. And biological systems have taken these minerals and stuck them in enzymes now. And so, it's a useful thing. It might have been originally part of our environment. The chambers are nice because it allows you to both concentrate resources and also have variation in the composition of those chambers that allows for sort of almost cell-like dynamics, right? You can have heritable variation at the level of a chamber. You have this proton gradient across the chamber, which is roughly the same magnitude as the proton gradients that we have in our cells now, and that's because you had these I believe, acidic oceans and basic water coming out of these hydrothermal vents, which then allows protons just to diffuse right down that concentration gradient. And, it seems to me that if you could get a rotary ATPase in there, you basically would just be able to make ATP for free. I'm not saying this is the origin of life, but like once you have something which can make a rotary ATPase, you have protons leaking through it as part of the environment, you basically have free energy. It's not that hard to imagine, and I'm still in the phase of imagining how this would occur, that you could have the conditions that are just a very fertile environment for any replicators that can couple environmental gradients of energy into their effectively metabolism, tying that energetic flow into replication, and then, you're off to the races. Certainly, I don't yet know myself. And again, I'm not working on the origin of life. I work on the evolution of multicellular organisms, which is in many ways an easier problem because we get to take for granted that living things have all these complex features already in a unicellular ancestor. But if you have something which has a ribosome and a rotary ATPase and, DNA, RNA, ribosome, like those are the basic building blocks of the modern life. And once you have those things, it seems like you're just kind of off to the races. How those exactly arose, I don't yet know, but I hope you bring on some good origin of life people and
Yeah, I mean,
hold their feet
that's
to the fire about it.
I mean, of course, I mean, the conversations that I had with the people who are working on origin of life, their response is the same, that they also don't know.
Okay. Yeah.
So, I mean, because of course it's difficult. I mean, Nick's argument of it kind of makes sense, but I mean, I wanted to have your views because it's important to understand that how you look at the problem, especially when you're thinking about multicellularity and kind of that minimalistic system, and what can be those barriers which are important across different life forms, etc.
Sure.
So, but with the origin of life, we get this kind of protocells, or I think this is what we call LUCA, right? Last universal common ancestor, or
those are legit cells. Like LUCA is a fully fledged prokaryotic organism. Protocells would have been a lot earlier, right? So you're talking things that are that are, probably in some sort of, there may have been some sort of ecologically generated, essentially vesicle-like dynamics, right? So Jack Stosak's done some really nice work showing that if you take certain types of um, polar and nonpolar chemicals, basically things that are a lot like the phospholipids that we have in our cells now, they will spontaneously assemble into vesicles. And if you, take they can basically have these sort of life cycle dynamics where, small micelles will sort of merge together, and you can have replicators inside those things, which actually increase their turgor by dividing, and that will osmotically bring in more components for them to divide. So if you have like a DNase or an RNase in there, it'll actually suck in free DNTPs and actually use them to polymerize their DNA or RNA, and that can actually then, if this thing gets sort of more turgid, it can actually attract more protocell material, which then causes it to grow more. These things don't look like the cells that we're used to that are nice and round. You get these strange tubular. They actually look kind of like mitochondria. They often look kind of branching and have these tubular connections. But you can get these sort of life cycles arising without having to have it under biological control. It's just sort of a consequence of chemistry. So when I think of protocells, I think of that. I think of something much simpler than something which has an organized life cycle, which controls the growth and reproduction of its cells. And certainly by the time you had LUCA, you kind of have, you have full-fledged prokaryotic organisms with much of the cell biology that the biosphere shares today.
Yeah, that's interesting, because then this also explains a continuum between protocells and something like LUCA, right? Because again, we'll have, or I don't know at which point we can say that, okay, this is equivalent to a kind of LUCA, or even modern bacteria. Like at this scale, we can say that, okay, now this is what we are looking for, right? Right.
Are you talking about the origin of life? Like where's
Like
the unanswered?
I'm talking about the transition from kind of this protocell
Mm-hmm,
to
mm-hmm,
LUCA, or to
mm,
a prokaryote, because this will be again a
yeah,
continuum, and
yeah.
it'll be really, really
Yep.
difficult to say that, okay, this is the, at this point now we can consider a cell as LUCA.
Totally. And I think, to be
Right.
clear, what I think is really remarkable about LUCA is that it is the deepest that we can trace sort of the lineage of all extant life on Earth. But if you imagine this as the sort of, this expanding frontier of different lineages, and you can trace it all back to sort of this origin of LUCA, we can't really go back any further in time, because any branches which branch off deeper than that last common ancestor, right? They're gone. We don't have any evidence of them, but they almost certainly existed, right? So LUCA was itself probably one member or one population. It doesn't have to be a single organism. It can be a population, right? But one sort of part of the biosphere. And there had probably been, a huge, large, complicated tree that precedes LUCA, right? And LUCA, so LUCA's, just to be clear for the listeners out there, I'm sure you know this, LUCA's not like the first prokaryotic organism which had sort of modern capabilities. It's just the last one that we can trace the lineage of life on Earth to now. But it would have been a member of a very large biosphere with a whole bunch of other things that are all very complicated, and sort of those modern lifelike capabilities had probably been around for a really long time by the time we got to LUCA. It's just that those lineages that were around simply didn't leave any ancestors that exist till the modern day. So if we had different sampling, if we had different preservation, we would have a different LUCA. And it doesn't mean that it would have had different features necessarily. It just means that we can either push that horizon deeper into the past and find something which has earlier branching, or maybe we would have actually pushed it closer to the present. And the LUCA that we know today wouldn't be the LUCA that we have. It'd be something that's more modern, because the LUCA that we know today would have been lost to extinction, along with all of the other biosphere that didn't persist to the modern era, or didn't leave descendants that persisted to the modern era.
Let's also briefly explain why we are talking about branching from LUCA.
Yeah. So, evolutionary biology is fundamentally it's a branching process, right? You have these evolution itself is it's a simple algorithm. You need something which has the potential to replicate, in which offspring vary. Some of that variation is heritable, and it affects their representation in the population in the future. Things that increase in frequency tend to win over those which decrease in frequency, and you have change over time. Once you have a system, a biosphere in which most of that reproduction is done in a vertical manner, meaning organisms reproduce the way that we know how they reproduce nowadays. They leave descendants of themselves. A single E. coli that divides, it leaves a daughter E. coli, right? A single dog that reproduces leaves another dog, right? You have this descent, which is in fact doesn't have a lot of information coming in from other sources. And as a result of that, as populations evolve, they can diverge. And we typically, and there can be breaks in the continuum of information flow amongst those individuals in a population. And that's typically what we call speciation. Speciation is the process of a population essentially having a lack of information flow between its members, such that now they're beginning to evolve independently. And if you do that long enough, it may be very difficult for information to ever flow between them again, because you get barriers to reproduction, barriers to mating. And so the result of that, that kind of vertical transmission, is that life itself is this extremely branching process, in which if you take all of the things that exist today, you can kind of go back in time and find common ancestors where there was a single population, a single species, which essentially left all of the descendants from that branch. So the common ancestor of all dogs today were wolves. I believe, Eurasian wolves, not American wolves, Eurasian wolves. The common ancestor of all mammals. You can go back further in time, and there will be a common ancestor of all modern mammals. You can go back further in time to the common ancestor of all animals 800 million years ago. You can go back further in time to the common ancestor of all eukaryotes. You can go back further in time to the common ancestor of all life. And that's what we call LUCA, the last universal common ancestor. But because of this process of reproduction, followed by essentially divergence of a population resulting in branching, if you play it forward in time, most lineages go extinct. Some of them, leave descendants which will diversify. And ecology, essentially evolutionary ecology, is understanding how and why these different lineages diversify and change through time. And there's some really wonderful model systems that give us insights into that. For example, the Rift Lake cichlids. We had one species of this fish populate these volcanic rift lakes in Africa, and now there's thousands, right? And they're basically occupying different ecological niches within those lakes, and they speciated as a result. So I will say the asterisk to this is that this is the way the biosphere works now. Most of the reproduction is vertical. Organisms reproduce a copy of themselves, and that copy is really similar to them. Prokaryotes, bacteria and archaea, do like to engage in a fair amount of horizontal transmission. Eukaryotes tend to suppress that, but prokaryotes like to bring in genetic information from other lineages. And so rather than this really nice sort of branching tree, where if you're playing it forward in time, you get further and further branching with things that are more and more distantly related to each other, it becomes more like a web, right? Where you can have things which, last shared a common ancestor in the vertical transmission sense a long time ago, sharing information between each other and making these horizontal connections in this tree of life. And there's good reason to think that pre-cellularization, it was completely different. Pre-cellularization, you wouldn't have had as much vertical transmission. You would have had essentially the components of metabolic pathways reproducing themselves, and sort of almost ecologically reproducing as a community, because their ability to get resources and divide is codependent on other parts of the pathways. But if you don't have nice cellular boundaries around these processes, the cellular boundaries allow everything to be really vertical, right? The cell divides, boom, everything is a copy of what was there before, right? It's the oldest form of vertical transmission, and it's a really effective one. It's why life is basically organized primarily around cells today. It's so effective. But pre-cells, it would have been really different. Instead of thinking of things as, a organism reproduces itself, right? And now you have a population of these things which are very similar. And, do those things vary? Sure. Is selection acting on that variation? Sure. Is the population changing? Sure. That's the way we think of evolution now. It's hard for me to even get my head around what it would be like if the subcellular components were their own replicators, and they were loosely coupled through ecological interactions, spatial structure on a landscape, how they're, inhabiting some surface environment, and the associations that they have in space almost provide that sort of organismal quality, but it's not like they're neatly bounded within cells. And it's a really different kind of evolution in that scenario, in that world.
Yeah,
People have
it's,
argued that allows for the fine tuning of the genetic code.
yeah, that'll be interesting to explore. So basically, what you have explained is that all the life forms that which are present or extinct species or extinct species, whatever species are out there or we can find out, all of them we trace onto this tree, which goes back to this LUCA, last universal common ancestor. But
Yes,
the
that's right.
first branching is between two major entities. One is prokaryotes, as you already mentioned, bacteria and archaea. The other one is eukaryotes, right? So what is this distinction between prokaryotes and eukaryotes?
It's a funny one, right? So we teach our students that we have three domains of life, right? We have bacteria, archaea, and eukaryotes. And the branching between bacteria and archaea is much deeper than that of eukaryotes. That's relatively modern, even though it happened on the order of 2 billion years ago. But eukaryotes are this funny thing, right? Eukaryotic cells are the cells that, if you remember back to your probably high school biology or maybe even earlier, right? Cells have a nucleus, they have a Golgi apparatus, they have vesicles, and inside the nucleus, they have their genome. Like, that's a eukaryotic cell. Prokaryotes, bacteria and archaea, aren't quite arranged like that, right? They don't have as much subcellular components. They don't have a distinct nucleus. They have a big circular chromosome, sometimes multiple ones. And it's all just, it's kind of more one pot for the cell as opposed to as many distinct compartments. Eukaryotic cells have been incredibly successful. They're an evolutionary singularity. There is, again, a last eukaryotic common ancestor, LUCA, not to be confused with LUCA, the last universal common ancestor. There's a last eukaryotic common ancestor. And this is one which resulted from one of those connections between diverse branches on the tree of life. This is one of those reticulations in the web, which actually became extremely important. So eukaryotes are the result of a symbiosis between archaea and bacteria. And so I think people are still trying to work out the sort of mechanisms underlying how this occurred and the timing, did mitochondria arise early, or did mitochondria sort of come into this more complicated cell much later? But at this point, it's really clear that the first eukaryotes arose as a result of an endosymbiosis where bacteria were living inside of an archaeal cell and then essentially became a part of the cell, right? So every one of your cells has, or maybe not every single one, I actually don't know humans very well, but most of your cells, that's true, have mitochondria that are these specialized subcellular components that provide ATP, the sort of energy currency of which cells work from. And those are actually bacterial descendants. They have their own bacterial genome. Most of their genes were actually shuttled off into the host genome, and they're sort of intimately controlled by your cells. They can't go off and live on their own. Their genome is shrunken, and they're really dependent on being a part of your cells. But this is a result of what we call an evolutionary transition in individuality, where previously there were two distinct biological species and distinct individuals, right? You had a archaeal cell and bacterial cell. And due to endosymbiosis and now the sort of coevolution of these two players together as one, they became merged into one species, one individual, and the bacteria became a part of the whole. It can't live on its own, right? And for most eukaryotes, it doesn't survive if you take their mitochondria out. And I think there's a debate in the field about how important mitochondria were to the runaway success of eukaryotes. Whether the energetic capacity afforded by mitochondria and sort of circumventing surface area constraints on a larger cell allows them to provide more energy or energy more efficiently to these bigger cells, which then allows larger genomes to evolve, which then paves the way, long down the road for more complex life. I'm actually not super, I'm not super convinced by those arguments, actually, or whether sort of what led to the ultimate runaway success of eukaryotes is just the plasticity and complex in cellular function that's afforded by having a lot more cellular compartments. These are bigger cells. They have a lot more sort of departmental functionality. They typically, if you're going to see cells, I mean, it's funny because there aren't that many absolutes in life, and it's hard to say the eukaryotes are absolutely better at any one thing. Bacteria and archaea are so diverse. They have the ability to move around quickly. They have a lot of the same abilities that eukaryotes do, and yet eukaryotes unquestionably have been terrifically successful. And most lineages of multicellular organisms that have evolved to be what we would consider really complex, so plants, animals, fungi, the seaweeds that you see washing up on the seashore, those all evolved within eukaryotic lineages. And there are plenty of multicellular bacterial species, but just all of them are quite a bit simpler than those which ultimately evolved in eukaryotes. And again, not every multicellular eukaryote is really complicated, but those that are really complicated, those multicellular forms that are really complicated, arose within eukaryotes. And one of the reasons that we think that might be the case is that eukaryotic cells have a lot of functionality. You have these, if we go back to the sort of, animal tree, right? Animals evolved roughly 800 million years ago, and they nest within this group called the holozones, which include relatives that aren't animals, like Choanoflagellates, ichthyosporians, Philisterians. And then if you go back even further, 1.5 billion years ago or so, you'll get a common ancestor, which includes fungi. And the holozones actually have they're these mostly like ocean living protists that have incredibly complicated life cycles. Now, we're not talking spatial complexity in the way that animals have all these cells that have differentiated themselves in space and you get a complicated organism. We're talking temporal complexity. The same lineage can be crawling around as an amoeba, swimming around with the flagella, aggregating to form multicellular groups. There's a lot of behavioral plasticity at the level of these genomes, at the level of these cells, that it's like becomes less a question of how do multicellular organisms invent these complex features, and more, how do they take these building blocks that exist in unicellular ancestors and start to arrange them for novel multicellular purposes.
Yeah, it'll be exciting to know especially once we start discussing about multicellularity, where this eukaryotic machinery can help in multicellularity. But before that, let's kind of talk about a bit of semantics here. So, how do we kind of understand or define, colonies, communities, and multicellularity in a way, because I think this is where it gets very interesting. Unicellular organisms, they can come together. I mean, we know bacterial colonies, etc.
Sure.
They
Yeah.
form, right? And
Yeah.
so how do you define it, and how do you kind of distinct between these three or more classes, if
I
there are?
honestly, I wish we had more words. I think one of the bigger problems in defining multicellularity is that different people use the same word to describe pretty different phenomena. They're all connected. In every case, something which you would describe as multicellular has, at a minimum, a group of cells. In a sense, that's an adjective. A multicellular group is just a group of more than one cell, right? When you say the word multicellular individual or multicellular organism or true multicellular organism, people start to add words that modify the adjective. And these things don't have universally agreed upon meaning. People are using it, and that's okay. And in fact, honestly, if you're submitting a paper in the topic and the field of multicellularity, just define what you mean by the term so everyone is clear. And I think you avoid 99% of the problems. Most of the problems occur when people interpret the word differently than you're intending it. And then they say, well, this isn't multicellular because it's not, my definition. Okay, so yeah, one of the challenges here is that multicellular organisms, multicellular morphologies, let's say, let's be agnostic here, multicellular things are extremely different from one another even just in extant multicellularity, right? We have everything that ranges from bacterial biofilms, which aren't, generally speaking, really very organismal. It's more like a community, to things which form aggregative multicellular groups under stressful conditions. So maybe they spend most of their lifetime as a single-celled amoeba. Food runs out, they aggregate to form a group. That group differentiates into something which maybe crawls around, looks for a good environment to sporulate, and then makes a sporangium. So there are these cellular slime molds, which dump a bunch of different species, the dictosteliids, which do that, and a bunch of others too. In fact, there are even ciliates which do this. They don't crawl around, but they form stalked sporangia. So that's a really common kind of thing, this aggregative thing. They live as single cells until times get tough, then they form a multicellular group. And then you have things which are, obligately multicellular, and they're clonal. So every group is a single genotype. They spend all of their life as a multicellular organism, except for maybe a very small reproductive bottleneck where there's a single cell phase. We're an example of this, right? We're obligately multicellular. We develop from a single fertilized egg. So every cell in our groups is clonal. The only time that we're not multicellular is at that reproductive stage, and that's really ephemeral, right? That's very different from something which spends maybe most of its life as a single cell thing and then has an ephemeral group stage. And so these are all multicellular. In a sense, they are the simple version of the adjective. They are all composed of more than one cell, but they're not the same kind of thing. They're not the same kind of biological organization. They don't display the same level of organismality and integration. And when I say organismality, what I mean is the co-functioning of the cells into a greater whole. Most biofilms aren't really very integrated and co-functional. You don't have cellular division of labor. You don't have reproductive specialization. Sometimes you get cool behaviors that sort of look like that, but it's probably not as controlled and regulated in the same way that we would expect a plant or a Volvox green algae to be, right? Where these things have basically been growing in obligately clonal groups, and natural selection has been acting on the properties of groups, and primarily groups, and pushing on those traits for hundreds of millions of years. And you get a lot of, essentially like altruistic behavior at the level of cells and integration, such that if you rip them apart, they're totally messed up, right? If you chop me into 100 pieces, I'm just dead. Right? If you chop a biofilm into 100 pieces, it's fine. So there's a lot of variation. They're all multicellular in the sense that there's more than one cell. So people have tried in different ways to sort of subdivide this variation. My preferred way to do it is to actually go with what philosophers like Peter Godfrey Smith have sort of taken a Darwinian population framework and applied that to multicellular populations. By the way, I'm happy calling anything that has more than one cell multicellular in the sort of small sense, in the adjective sense. It is more than one cell, no problem. You have a multicellular bacterium because it forms groups, that's fine. We can then level it up one more. We can say, is it a multicellular individual? And by that, I mean, is it a multicellular Darwinian individual? Is it at the group level an evolutionary unit, which has the ability to gain adaptations? That's actually, I think, to my mind, that's really fundamental, because, a lot of the features that we care about in terms of multicellularity is this sort of coordinated, refined, multicellular level complexity. We frankly wouldn't be talking about this if multicellular was just groups of cells, right? It's what we care about it because they are complex. The word complex itself is, another can of worms, which we don't need to get into. But we care about multicellular organisms because they are units, evolutionary units, which have the ability to gain adaptations. And if you accumulate enough of those, you can get fantastically complicated organisms out of that process. Animals, plants, mushrooms, those are all independent examples of that happening. We know in every case, we start out this process with a very simple clump of cells. And you go hundreds of millions of years down the road and you have a redwood tree. It's a big difference. And that's why I care about it, right? So in every case, we have this sort of evolutionary framework where you start out with something which forms a group, and there's a life cycle. Groups are able to beget new groups. Groups reproduce somehow. It could go through single cells which form groups. It could rip itself in half. It matters in the sense that those details do end up being important. But on the other hand, it doesn't matter that much. What really matters is that groups can create new groups. And then you need, variation in group level properties. That variation has to be heritable. An important part of that is getting genetic bottlenecks at the level of group formation, such that natural selection acting on whole groups is affecting the frequency of the genes in those at the population level. If that's not the case, then natural selection acting on groups doesn't do anything. You need to have groups be different from the population as a whole. And so, that, and also having essentially having genetic bottlenecks at the level of groups also helps group level traits become heritable. If group level traits are heritable and selection's acting upon it, then you're going to get group level selection driving group adaptation. And if you accumulate that over time, you can get organisms out of it. So if you do the work to show that something satisfies Darwin's criterion for a Darwinian population, or Lewontin had this really nice paper in the 70s talking about sort of Darwinian criterion, you can do that for any multicellular, population. You can show that groups beget groups, that there's heritable variation in multicellular traits, there's genetic bottlenecks, and that selection acting on those groups can drive evolutionary change, right? At that point, those things are multicellular Darwinian individuals, or multicellular individuals for short. Not that many people do that kind of work. That's the kind of stuff we do in our lab. It's sort of a pain to do, and it's very difficult, if not impossible, to do with natural populations, because you don't know what the environment is in which you can actually assess some of these things. So there, I think it's fine to just show group reproduction and bottlenecks and heritability of group traits, and then we can leave selection out of it, because that can be very difficult to measure in practice for natural populations. Pretty easy in laboratory populations, very difficult natural populations. And then the highest sort of criterion would be multicellular organismality. An organism is something which is defined by sort of Dave Queller and Joan Strassman defined it as something which has high cooperation amongst its parts and low conflict. And I like that, right? Because you can actually have organisms. Organisms in some ways are the functional consequence of multicellular Darwinian individuals. Enough selection on evolving groups can give you things that have division of labor, things that have, essentially more, multicellular development, things that have sort of the features of multicellularity that allow them to be better multicellular organisms, that allow them to have higher fitness as groups, but also make them more complex as a result, right? Our cells becoming interdependent, such that if you take them out of the group, they no longer do so anymore. Our groups breaking trade-offs that single cells face. Single cells face all sorts of trade-offs. Can you grow and reproduce at the same time? Some of them can't. Oh, in a multicellular group, sure, these cells grow, these cells reproduce. It's problem solved, right? Are there trade-offs about specialization, right? Would it be better for half your cells to specialize on one behavior and the other half specialize on the other, versus everybody being a generalist? There's all sorts of scenarios where that's true. A multicellular organism, you can take advantage of that, right? So organismality is sort of that next step, right? If you have a lot of group adaptation, you can then get to the level of organismality. So those are, I think, three different ways of describing any multicellular system. Is it group forming? That's multicellular. Is it a Darwinian unit? That's an individual. And does it have functional integration? Well, that's an organism.
Yeah, this is really exciting. I think, like, just to understand, and especially when once we start or try to define all the different things at cellular level, or really at the basic level, we see that many of these things they start to break down. I remember the debate between the selection working at the group level, or
Oh yeah, that
because
old
this was
chestnut.
like this was a major debate in 20th century. So
It's a
where
funny one. It's a funny one too. I'm delighted that that's largely died off.
yeah.
When I was a graduate student, it hadn't. It was still super fierce. But even at the, I came up in a very group selectionist a very kin selectionist anti-group selectionist lab. And I actually remember at a conference once like gasping, oh, but that sounds like group selection. Like, and I think I said it out loud. I was like, I can't believe it. It was heretical to invoke group selection. And I get that there was some really bad group selection models out there. And the main issue, with a lot of these bad group selection models is the lack of strong bottlenecks at the level of group formation. If you have strong bottlenecks at the level of group formation, I think everybody kind of at that point is like, okay, like if groups are clones, then well, the kin selectionists don't have any problem because like the relatedness between cells in the group are 100%, and so sure, it doesn't matter if a bunch of them give up their ability to reproduce. If that means that they reproduce more as a clone, the math pays off, right? But the funny thing about that is that, well, I think we should probably avoid this tangled topic.
Topic.
And largely speaking, it is gone away as a major source of contention within evolutionary biology. And I will say that there has been a lot of sort of ink spilled over this, but most people would kind of agree that both frameworks are reconcilable because they both fundamentally function through the same mechanics of, there's only one there's only one reality of there are things that are reproducing that are Darwinian, and you can look at it through a lens of, I'm going to keep track of how these entities are related and how their behaviors affect their own reproduction as well as the reproduction of relatives. Or I can think about it as, I'm going to think about how natural selection is playing out on different levels, and I'm going to start analyze it from that perspective. Which perspective you take, whether I'm going to only think about like one level, like only cells, and cells live in groups, sure, but I'm just going to think about how cells are reproducing in the context of groups, and how they're related to those that they're interacting with. Or I'm going to think about sort of selection acting at separate levels, right? And I'm going to try to account for that in terms of how this let's say a genotype is changing in frequency in a population. They're both looking at the same thing. There is only one reality, and there's different ways to look at it. And sometimes it makes more sense to use a sort of, inclusive fitness approach. Sometimes it's just simpler to use a multi-level selection approach. For example, I would hate to try to explain how selection acting on the properties of a multicellular organism are really acting on cell level properties.
Yeah.
Now, let's say that let's say that the trait that I care about is, I don't know, a mating dance of a bird. Like, can you write a model that explains that from the perspective of the cells in the bird? There is no like cell level trait that corresponds to the bird's behavior. It's a multicellular trait that has, of course, it depends critically on the underlying properties of the cells, but it's not like we're talking about something where the selection's acting on the group, but really the trait is a cell level trait. Like, what proportion of the cells are cooperators? What proportion of the cells are producing a compound? So when you get into these, like if you have a group and the group has traits that don't have a clear mapping onto cell level traits, then everyone, even those hardcore kin selectionists, just shift to a multi-level selection approach, and they just say, okay, well, that group's an individual. But it's an individual, but it's also a group, right? And so everybody always gestalt switches, even if they kind of argue that they don't. And so I don't know. I've
I
got
think,
complex feelings on this problem.
yeah, I think a lot of this thinking comes from our reductionist approach, right? Like we want to try to explain everything from like at certain level, but we kind of, I mean, I think now a lot of people, a lot of scientists, they are appreciating the emergent phenomena, emergent behavior in general in science, right? Because I think some explanations you can't like dissect any further. One example that you gave, like the mating dance or something, and you can't really explain it at cellular level. At
I mean,
cellular level, I don't know
I
how
think
much
in
of it.
principle, you have to be able to. Like, I don't think there's any like magic that like comes in and makes that non-explainable from a lower level phenomenological perspective. But it's incredibly hard to do. It's incredibly, the amount of information you would have to have about the group is ridiculous. And we're not going to have that much information. Maybe so for some questions we will. For some questions it becomes really, really, really hard, and so it's just become so much easier to move up a level, right? And I
So
think in the case of the mating dance, I'm sure there's a lot of emergence happening, but I think a lot of it too is actually the consequence of biological mechanism. You know, Like for the mating dance, I think I would probably argue less of it as emergence in the sense that it's just sort of comes into being fortuitously and hey, it's useful. And more, it's the result of a lot of biological adaptations which allow for neuron systems and intricate morphological innovation and eyes and communication and memory and instincts and this contingent on brains, and there's so much building of the organism that has to occur to have that. And in some of these cases, you might even argue that a lot of that building is because of the benefit of some of these traits, right? Being able to succeed in mating, is a very, very valuable thing in evolutionary biology. A lot of natural selection is about not just being stronger and living longer, but actually being the one that reproduces, and often at great cost to living longer, because it's just such an important component of fitness. Anyway, so I love emergence, and I think emergence is really often really easy to see. So I think this is true for all levels of biology. I think emergence is often really easy to see when you're in the relatively early stages of a new way of doing things. And you don't have a lot of biological mechanism and complex adaptations that are really good at getting you to somewhere being layered over this. So if, for example, in the evolution of multicellularity, early on, when you have groups that arise from single cells, those groups may provide a benefit that is fundamentally different from like there may be a group level trait which is adaptive, which is fundamentally different from the cell level trait that it depends upon. The swimming speed of a group of cells may depend on the shape of that group. And the shape of that group is going to depend on something about the cell cycle of the cells that affects the shape to which they divide and how they assort themselves in space. So you get a mutation, which changes the behavior of the cell cycle, that changes the shape of the group. All of a sudden, the group is swimming faster, and natural selection cares about that, and it's acting on fast swimming. So selection is acting on a multicellular trait that, of course, has to have an underlying mechanism. Otherwise, it's not heritable. Otherwise, it doesn't matter, right? Otherwise, it doesn't matter in the sense that it can't progressively change through time. It has to have a mechanism which is heritable, or I don't really care about it. Let's put it that way. And it probably won't matter for the long run. But that mechanism can be fundamentally different. Cell shape, division plane at the cell level is different from group shape and swimming speed at the multicellular level, even though there is a connection between the two. And I would argue that the multicellular thing there is kind of an example of emergence, right? There's a mutation, it changes the way the cells divide, boom, all of a sudden these groups are growing differently, and it really matters to their reproductive success, their survival and reproduction. It matters to their fitness. And you can kind of see emergence happening in this fortuitous way in really simple systems because they don't really have much obscuring complexity yet. But if you go 100 million years down the road, right, and now all of a sudden you have these morphology programs coordinated by the expression of morphogens and age-dependent signals that cells are doing, and they're making it really, really sophisticated and robust, kind of like the way enzymes take things that happen spontaneously, make them happen a whole heck of a lot faster and better. That's what development does too, right? Is it basically gets you there more robustly, more efficiently, in a much better way. But now the emergent part of it is like kind of hidden because you've layered a bunch of biological mechanism over what was originally just kind of given to you for free. I think that happens all the time, right? It probably happens at the origin of life, where you get life cycles that are free. You get things that are basically, polymers that are growing and splitting in half, and all of a sudden there's two of them, right? Or protocells that are extending and pinching off, and all of a sudden you have two of them, right? These things start out essentially creating a closed Darwinian loop. They start out with something which allows for Darwinian processes to play out. And it's kind of emergent. It's kind of not, it didn't evolve to be that way. It's just a consequence of their environment and physics. And then that allows biological innovation. That's the magic of life. It allows essentially this whole process to become more regulated, more fine-tuned, more robust, better. But at that point, the emergent part of it, even if that was the germ, it's hidden because it's under a bunch of layers of biology. So I think emergence is profoundly important. And where you probably see it most clearly is in those very early stages of organisms doing something new, where they don't yet have the opportunity to put it under a lot of, elegant regulation.
So basically, you think about emergent in a way like it's a data problem. That means if we have enough data points, we'll be able to explain any emergent phenomena. Or because you said that there is nothing extra
Mm,
happens, right? But we can kind of look at it in
mm,
abstract way. I mean, looking at it,
mm.
even, behavior of slime molds and stuff like that, right? Like
Yeah.
it is pretty complex. I mean, because I think what you're trying to say is that at some point we'll be able to, or I don't know, or at least hypothetically we can explain psychology from whatever is going on at the cellular level, which
Harry
is
Seldon, psychohistory.
uh,
um, I mean, this is, I think, maybe more of a philosophical question for me than a scientific question.
yeah.
Um, I'm not a philosopher, but I've been more persuaded by the people that argue that strong emergence, like emergence that is effectively fully decoupled from the lower level causation, isn't kind of isn't real. Like it's more a reflection of our lack of understanding of how the system works than of how the world works. That, like, if we're talking about sort of hierarchical levels, right? Behaviors and traits that are manifest at one level, which of course depend on lower level things, right? The mating dance of I don't know, I have chickens in my backyard. So the mating dance of a chicken um, clearly depends on the cells in that organism, right? It arises as a consequence of the way that cells behave. Brain cells, muscle cells, feather cells, all those, well, feathers aren't cells, but you know what I mean. The cells of the organism are certainly the causal players in the behavior of the organism as a whole. Um, but I don't think there's any sort of like mysterious gain of function between those two that couldn't potentially be explained by a very by an accurate, a very accurate model at the lower level, which basically tried to explain this phenomena in terms of what every cell is doing, its inputs, its outputs, its state. Um, and I guess I do work on. I've thought about this much more in the context of multicellularity than any other context in biology. And again, I work on those really early stages where a lot of things are fortuitous. They start out for free. Life cycles, you get them for free. Heritability of group level traits, you get it for free. Novel multicellular traits that don't correspond, that depend upon, but are not corresponding to cell level traits, you get it for free. Can you get runaway multicellular adaptation? Absolutely, right? And so for me, I think that type of emergence where you basically have something which functions in a domain where it's never had to function before, and it's actually okay at it. It can do it, even though it's not evolved for that purpose. To me, that's a level of like simple emergence, right? You have a group. That group has properties at the group level that that you get essentially not because they're adaptations, but because that's just the way that physics works. It's just the way the world works. But there's something which you can kind of like which natural selection grabs into and says like that actually matters. Like let's pull on that. And I think a lot of the behaviors that we typically care about in other multicellular organisms likely started the same way. But at this point, it's less about sort of things that kind of arise without causation and nonetheless are useful, or not without causation, arise not because that lineage had evolved to do that, because doing that was beneficial. The mating dance clearly is an adaptation. The mating dance evolved because it is beneficial. And it may depend on all sorts of emergent properties at various levels. That's just the way organisms work, right? There probably is all sorts of hooking into emergence, but like that behavior itself is almost certainly an adaptation, right? But anyway, yeah, I typically think about this in simpler contexts where you can see the emergence, and you kind of know that like, hey, this thing, it's either it hasn't been multicellular in 250 million years or ever. All of a sudden we make it multicellular. All of a sudden it has new multicellular traits, which are good. And all of a sudden natural selection starts to act on those, right? So that's a kind of a different type of emergence. That's sort of like the emergence of gaining functions, not because it was adaptive to do so, but simply because that's just the way that things work when you go between one hierarchical level and another, and all of a sudden being able to evolve those functions and leverage that emergence for novel, for novel purposes. But so yeah, my to answer your original question, my reading of the philosophy on this and my mild preference, mild to strong preference is that all emergence is weak emergence, even if we can't explain it yet. But I don't see any reason why there would be some sort of jump from one level to another that is so much more than the sum of the lower level, or maybe a complex mathematical function of the lower level parts, not necessarily a sum, but some function of the lower level parts that can give rise to it, because it does arise from those lower level parts. It certainly depends on those lower level parts, and it must come about because of those lower level parts. And I don't think that there's a jump, a mystery in that gap that cannot be eventually explained, even by nitty gritty reductionist approaches. It would just be unfathomably difficult to do so.
Yeah, I mean, in my conversation with Nick Lane, he mentioned that it's basically integration of different systems that coming together, and that gives like new emergent phenomena or something.
That certainly is true. Yeah.
Yeah. Which, I mean, of course, I think once we start talking about this in the context of your work on, especially on Snowflake yeasts, let's start with that. What are those, and like the kind of experiments that you are doing, and then we can come back to emergent phenomena.
Awesome. And I'll try and bring it into the description a little bit.
Yeah.
All right. So about a decade ago, I was having a conversation with with a collaborator who became my postdoc advisor, Mike Trevisano. We were talking about like, what's the coolest, work we could do? We were like, origin of life, that would be the coolest. And that's literally the conversation we had. We both were like, yeah, but we're not chemists. We are biologists, and we don't know enough chemistry to really make progress there. And I'm glad we had enough self-awareness to realize that. Like, what's the second coolest thing we could work on? Multicellularity. There we go. So, we sort of know from the comparative, we have sort of four major ways to study multicellularity. We have the fossil record, we have comparative biology, all these extant multicellular organisms which differ, and we can examine their traits and put them in context and try to learn how they gain those traits, and et cetera. And then, we can do theory and computation. We can sort of do first principles logical arguments about how things work. And the fourth is that we can just evolve multicellular organisms completely from scratch and learn how this process works. One of the really nice things about mixing all four of these approaches, we really do the latter two, the evolving it from scratch and theory and computation. But one of the nice things about having all four is that the only truly long-term experiments that we'll ever have access to are those which have already happened on Earth. We have a large number, at least 50 different things, which have spent hundreds of millions of years evolving multicellularity. We are never going to catch up to something which has 100 million year head start in the lab. It's so far out there, which is really cool. So we can see what happens over long time periods. But at the same time, we know that a lot of these we know that multicellularity evolves repeatedly. That means it's relatively easy. If it was really hard, it wouldn't have happened 50 times. We know that in virtually every case, multicellular organisms aren't inventing new genes and pathways. For the most part, they're co-opting things that already existed, putting them to novel function. And so what I really want to understand are the dynamics of how that occurs. I want to understand how a simple group of cells arises and then actually begins to do that co-option, begins to actually evolve into a integrated, more complex organism. And there's a bunch of sort of questions there, right? Like, how do early multicellular life cycles arise? And how do the properties of those life cycles affect what happens downstream? How do groups evolve to become biophysically tough? We tend to sort of take this for granted, but when group multicellular organisms start out, like, they've never had to deal with the stresses that act over long length scales. They've only dealt with cell level stresses. Now all of a sudden they have to make a tough body. How do they do that? how do you get, cellular differentiation, division of labor, cell type specialization? That's a huge one, right? The origin of differentiation within and development within multicellular organisms is really something which we don't have a good dynamical understanding of how that actually occurs. We sort of know how it has evolved, what genetic pathways, what processes in different lineages, but we don't really know how you get from not a non-developing group to something that has development. How do environments affect this transition? I mean, there's a whole bunch of other questions that I'm interested in. And it's really about sort of, not necessarily what can happen over the very long time, but the process, the evolution. How does this occur? And so that's what we set out to do. We're doing currently a long-term evolution experiment in our group. We're on generation 7,000 of what I hope to be a 50,000 generation experiment. I want to keep doing this until I retire in maybe 30 years or so. And in our lab, the long-term experiment is led by a research scientist in my group, Ozan Bozdag, who's a fantastic scientist and had both the vision and wherewithal to do this. It's something which requires that you're in the lab 7 days a week, 365 days a year to do the selections and do the transfers. And so it's a tough experiment. It's an endurance race. We decided to work with yeast because yeast are a great model system for all sorts of cell biology. You can do synthetic biology very easily in them. They're easy to genetically modify and confirm what different mutations are doing. They're easy to sequence. They're easy to grow. They're just a great model organism. And we started out with a single-celled yeast. In nature, we know that the first step is that, there's an advantage to group formation. And so cells evolve to form simple groups. We just published a review paper last year looking at 10 different ways, 10 different selective drivers of simple multicellularity. There's no one, I only say that because there's no one way that groups form. There's a lot of different ways that groups form, and for different reasons. They often kind of come down to increased survival in the face of stressful conditions, increased motility, or increased metabolic efficiency. They're doing some sort of metabolic process, and it's good to concentrate it in a group of cells. Actually, not that different from protocell formation. So in our case, instead of trying to apply any of those natural selection conditions, although we have done experiments with predators and we get similar results, they all have downsides. We do the simplest thing possible, which is we grow our yeast, and every day their number expands 100-fold. We have about 1 billion cells in each population. Then we put them through a race to the bottom of the test tube. Importantly, we only put a subsample of the population. 10% gets to race to the bottom of a test tube. We put them on the bench and just let them sit for a few minutes. The time which we allow them to sit depends on how fast they're sinking. Groups sink faster than single cells. We take the bottom of the ones that make it to the bottom of the test tube. They go back to fresh media. Everything else is killed, and you rinse and repeat. It's a really simple experiment. And for our listeners, if anyone's going to do their own selection experiments, my advice after doing this for a while is keep it really simple. Complicated experiments either fail or go sideways on you, and the organisms do something really weird that you'd have never predicted, but in hindsight, it makes sense that they would do that. Keep it simple, more likely to succeed. And we've done that every day for 1,400 days so far. We get about, that's about 7,000 generations, cell level generations. Okay, so what happens? Well, initially, I should well, I'm going to keep this brief because I'll take way too long if I make this like a full-on faculty, shock talk. We get a life cycle for free. It turns out that our yeasts grow with a beautiful branching structure, which is why we call them snowflake yeast. They look sort of like snowflakes. They have this beautiful branching structure because daughter cells no longer separate from their mother cells. That's on account of one mutation that makes them form groups. At this point, they are little m multicellular. They have multiple cells per group, but they're dumb clumps of cells. Are they multicellular individuals? Well, it turns out that they basically grow until they hit a certain sort of size. If you imagine that you're building something which looks like a tree from your backyard with ping pong balls, and you're adding one ping pong ball onto each one that exists every time the cells divide, pretty soon the center of your little ping pong ball statue is going to get really densely packed. Try and jam another ping pong ball in there. What do you think happens? It breaks in half, right? Our yeast start out with pretty round cells, so they grow to be about 100 cells before the jamming gets to be really bad. That jamming then breaks these things in half. When they break in half, essentially, if they break any cell-cell connection, It's like cutting a branch off a tree. That cell and everything downstream falls off. So we have this emergent life cycle. It's emergent in the sense that it didn't evolve for a reason. It's just there from the very beginning as an outcome of the way that groups form. So we get these trees that form three-dimensional sort of trees. They're growing in all directions. Then they have a life cycle. So now you take one snowflake cluster, you put it in a tube of media, come back at the end of the day, there's a million of them. They're all the same size because they grow to be that. Essentially, they grow until that packing causes fracture. Great. Turns out that because of this, there are these fantastic bottlenecks in the structure. So every time it breaks, right, it's broken off of a single cell breaks, and then the rest of the branch falls off with it. The rest of that branch, though, are its daughters, its granddaughters, its great-granddaughters, and they're all clonally related to it. So when mutations happen, which they do, whole branches get that mutation. And then when that chimeric group fractures, every time it fractures, it releases a clonal offspring. Whether it's 100% wild type or 100% mutant, every time it breaks, it will be guaranteed a clone. So if you take one chimeric cluster and put it into a tube and you come back at the end of the day, you have a million clusters. 999,000 are clones. One of them is chimeric at best. So mutations segregate between groups. And if those mutations create new emergent multicellular traits, natural selection sees it, it grabs it, pushes them to high frequency, everyone else goes extinct. So what happens over the long run? Well, first thing is our cells start to evolve more and more elongate cells. Going back to your model of ping pong balls, right? You're adding ping pong balls to the tree. What if instead of ping pong balls, they were hot dogs? Same structure, it's a branching structure, but now all of a sudden it's super fluffy. It's taking up a lot more space, right? You have a lot more free space around those cells. They're not jamming up, they're not breaking. So you can get something which the longer the cells are, the bigger this thing grows before it breaks. And what we see is basically these things get bigger and bigger and bigger and bigger and bigger up to a certain point when all of a sudden getting a little bit more elongated. We're talking about cells that went from being almost perfect spheres to being, 2.5 times longer than they are wide. Now, all of a sudden, instead of being a little bit bigger with more elongated cells, they are 10,000 times bigger. We're talking about our groups going from 100 cells to 1,000,000 cells, still being clonal, but 1,000,000 cells. If we look at their toughness, they're going from the strength of actually 100 times weaker than Jello to as strong and tough as wood. How the heck are they doing that? You can't strengthen one cell-cell bond by 10,000-fold. They do strengthen them by about 6-fold, not 10,000-fold. There, it's actually it's a change in the way that these cells are interacting. They're actually going from a branching growth form to one which is entangled. The cells basically wrap around each other like ivy wraps around, trees. I live in the South. I'm often ripping ivy off of trees. It's ridiculously hard to pull because of that entanglement. If you pull on one bond, it pulls on 5 bonds. Those pull on 5 bonds, and that percolates so that to rip this thing in half, you have to rip apart thousands of bonds, and it becomes thousands of times tougher. These are all emergent properties at the level of the group. I should say one of the major stumbling blocks in our system is that, or in multicellularity in general, is that people really think that the emergence of heritability, what makes group level traits heritable, requires explanation. It requires the creation of some new layer of biological regulation. For example, if you have genetically regulated development, now you have a part in your genome that you get a mutation, and now that affects the way three-dimensional structure builds. That is heritable because it's based on that single mutation. Now we have a genetic basis for a multicellular trait. Snowflake yeasts don't have that. They're dumb clumps of cells. And yet, when we actually measure the heritability of group level traits, it's higher than that of cell level traits. Considerably higher. That's really surprising. We didn't expect that it would be, but it actually, in hindsight, that these emergent traits, like size to which they grow before they break, are more heritable than the cell level traits that they depend on, like the shape of the cell, mutations affecting the cell cycle that affect the shape of the cell. It makes sense because these emergent multicellular traits actually get to average over. They sort of depend on ensemble properties. They get to average over some of the noise at the cell level. So even if this trait is noisily expressed at the cell level, the group actually gets to cancel out some of that variation. And so the group level traits have higher fidelity to the mutations than the cell level traits that are directly resulting from the mutation itself. Long story short, we're also getting cellular differentiation arising, we believe. We've done single cell RNA-seq, and we go from having one cell type to 3. One of them we believe is involved in making some of the cells that are at the base of a bunch of branch connections really strong. They appear to be overexpressing cell wall biogenesis genes. And the other one appears to be essentially involved in creating refining the multicellular life cycle. There's a lot to get into. I know I could spend hours talking about our system, but writ large, I'll just summarize. We did the simplest experiment you could imagine. Actually, the second simplest. The simplest is a wonderful system that Rich Lenski pioneered, evolving E. coli for 75,000 generations for faster growth. We basically did that. We're doing that. We're on 7,000 generations, not 75,000, but with this extra step of sedimentation. Again, we're working in a eukaryote yeast, not a prokaryote E. coli. These group-forming snowflake yeast evolved really quickly. They have an emergent life cycle in which they grow until they hit a certain size, and they start popping off babies, which then grow to the mom's size. They start popping off their own babies, and you get a population of groups. Mutations arise in this context and are segregated out into their own groups. And those mutations create new multicellular emergent properties. You change the shape of a cell, you change the size to which the group can grow, you change its biophysical toughness. Natural selection cares about that and acts upon it. And what we see over thousands of generations is the evolution of changes in cell morphology, make the cells a lot longer, changes in group morphology, our groups get 20,000 times bigger and as tough as wood. And we think, we haven't published this paper yet, but it's work that we're actively working on, we're seeing the beginnings of cellular specialization. We're doing a lot more stuff too, but that's sort of the big picture of our long-term evolution line, and it'll be really cool to see what happens because I keep being surprised by the system. I can't really necessarily predict what will happen, and that's one of the real joys of it.
Yeah, it is like really, really amazing to know all this. But did you ever discuss with Richard Lenski about this, like whether he kind of also observed something similar with bacteria, or if it
Well,
can happen?
he never incentivized them to form groups. So
Uh-huh.
he didn't do the step where you would select for group formation and
Mhm.
reward larger groups rather than smaller groups. Had he done that, I'm not sure what he would have found. So E. coli have a specific one of the things that really matters here is like the interplay between the ancestor cell biology and how they form groups. So if you take an E. coli, which is rod shaped, it undergoes binary fission, where this rod splits into two rods, right? Pretend these are rods. And so if you basically select for group formation, the first thing you'll get are filaments, which are kind of common in bacteria, right? These things that no longer divide from their mother cells. And they form basically, long chains of cells. He could, I mean, I suspect that that's what would happen if you were to do this with E. coli. They might make biofilms, they might secrete sticky proteins or extracellular matrix such that they form clumps. Our snowflake yeast don't do any of that. The only connections they ever form between cells are between mother and daughter cells. And they're not sticky. Our groups truly are clonal under experimental conditions. And that's actually, it's not necessary for multicellularity to have clonal groups. There are all sorts of multicellular organisms that are not purely clonal, but it makes life a lot easier for them to be clonal. Because basically, if a group has one shared genotype, if there are mutations which affect emergent group level traits, then selection on the group reflects really well on that mutation itself. If the group contains multiple different genotypes from the population, then selection on the group is affecting multiple different genotypes at the same time, which makes it harder for there to be this correspondence between gene and group trait. And it also means that it might be harder to recapitulate the emergence of that group level trait. It might depend on the interaction between a bunch of different members. And if you pair them up with others, you don't get the same trait. So there may be maybe noisier expression of the emergent trait, and there's not as much fidelity between a group trait and a genotype. So clonal multicellularity is just like, it's really easy. And there's the kind of a reason, I think, that it helps explain why all of the lineages of what we would consider complex multicellularity are clonal. Plants, animals, fungi, seaweeds, they're all clonal. And that's not the only reason, but it's probably one of them.
Yeah, and can you please briefly mention about the reproduction in yeasts, because I think that will also
Yeah.
matter, because I think there are again different methods to do it. So,
Yeah.
so yeah.
Totally. So budding yeast have a specific type of reproduction, right? You have a cell, and it's basically pops off a daughter bud. Normally, in diploid yeast, actually, they alternate which pole they get the bud. One to the other, one to the other. Which, if you're imagining that, you have one cell, it's got a daughter, then you push one back, then you push forward, then you push back. If every cell is pushing back every other division, there's a lot of crowding on the middle of that thing. It's not going to get very big. We got really lucky that the mutation which causes them to essentially not create the enzymes which digest the cell wall connecting a mother and daughter cell, which allows them to form these permanent bonds between cells. They're connected with a chitinous ring by their cell wall. We got lucky that that also messes with the bud localization checkpoints. So they no longer back bud. So now they're basically they're growing outwards like a tree, where every time they bud, they're adding another cell to the outside. Every cell is adding another cell further away. And you have one thing which now forms, I mean, you can look on our website and see cool pictures, but like these things are basically growing outwards away from a common core in 3 dimensions, but they look a lot like a little, I don't know, marine organism or something. They have this sort of, coordinated branching growth away from a common core. That's just the consequence of, we kind of got lucky, right? Like that's just cell biology, the cell biology of the ancestor, and a mutation that messes with it. We have some unpublished work in the fission yeast, Schizosaccharomyces pombe, which shares a common ancestor with budding yeast hundreds of millions of years ago, and has a really different type of budding. Instead of reproduction, instead of budding a daughter cell, so in snowflakes, if you have like 5 divisions, right, from one cell, it will have the oldest cell will have 5 branches coming off of it because it's budded 5 times. The first branch will have 2 to the 5 cells connected to it. The second branch will have 2 to the 4. The third branch will have 2 to the 3, 2 to the 2, 2 to the 1, right? So 1.
Yeah.
Or 2 to the 0. Sorry. 2 to the 1, 2 to the 0. So that would be 6 to 3. But you get my point. You get this cool sort of, binomial expansion where the first branch is half of the total biomass of the group. It's got a nice little architecture. This other yeast splits its body in half. And so it's got a totally different mechanism of division. We evolved it. Ozon and Agrasidium lava rosen evolved it. And it actually so they started out actually with a spherical mutant so that it could divide in multiple different planes and not be, sorry, I don't want to get into the backstory for that. But we started with a spherical mutant and evolved it for settling selection. And it got the same mutation as the snowflakes, ACE2 deletion, this mutation that prevents daughter cells from separating from mother cells. But now it creates what we call cuboidal packing topology. Specifically, snowflakes have this thing where the oldest cell has a bunch of branches, the second oldest cell has one fewer branches, the third oldest cell has 2 fewer branches than the oldest cell. This really nice regulated network, right? This thing, it starts out with 1 cell. Same mutation, right? Daughter cells stay attached to their mother cells because they share a cell wall. Now what happens is it splits like this, and now these 2 cells are sort of like squished because they're stuck to each other. So now the next division plane is 90 degrees relative to that. And now you have 4 cells, like a little hot cross bun. But actually, if you look at it in this direction, they're kind of squished and tall in this direction. So the next division tends to occur in this plane. So you get little groups of 8 cells forming a cube. And if you let those guys continue to grow, you get groups that have these cubically packed modules. That's an extremely common form of simple multicellularity. We see it in archaea. We see it in bacteria. We see it all over eukaryotes. I mean, we see it in 600 million year old fossils from the Ediacaran, sort of the origin of animals. Nobody knows what they are. I think they're algae. Nobody knows what they are. You even see them in things that look like they have like lamellar sheets of cells with these cubically packed things along the way, which look like sort of simple algae with blades, with blades of a body that still have packing geometry that's this cuboidal geometry. And so Rosen is basically showing that, in fact, you get, much like snowflake yeast, you get a life cycle for free. You get these cubically packed groups which grow and pop off little baby cubes. Every baby cube has 1 cell at its base. If there's 8 cells there, well, they shared 1 common ancestor, 3 generations ago. So you get genetic variation being segregated in space. They're evolvable. I mean, they're basically these little multicellular Darwinian critters. They're very different from snowflakes. They have they're still members of the same general topology class. Snowflake has something where, older nodes can be attached to multiple edges. These ones do too, but they have a very specific packing that results. It's very, very different. And actually, if you take the same sort of basic cell biology, but instead of having these division planes that rotate, you make them parallel, you just get a chain of cells, right? You get a filament. If every time you have binary fission, but the division planes are always parallel, you just get something which grows forever as a single filament. So filaments, cuboidal packs, snowflakes, they're all very similar. They're all united by the same fundamental topology class. And they segregate genetic variation pretty well. They serve as a substrate for Darwinian individuality, for multicellular individuality. And they do it through this process of emergence. You get physically, I don't know about filaments. I haven't investigated those myself, and I don't know if anyone else has done these experiments. Typically speaking, our field is really small, and so it's not like not like a lot of other people are doing these experiments. but at least with snowflakes and with cuboidal packing, you get a life cycle that arises as a consequence of emergent physical packing. And that life cycle includes very strong genetic bottlenecks, almost perfect genetic. They're not the best they could possibly be, but they're very close to being as good as they could possibly be. Great genetic bottlenecks, which allows group level traits to become heritable and sort of facilitates the ability for natural selection to act on group traits and to make multicellular groups, multicellular Darwinian individuals, and then over time, possibly multicellular organisms. That's really where I'd like our work to go. But it's easy to show multicellular individuality. To get organisms, that just takes time. It takes a lot of evolutionary change.
Yeah, so I mean, of course, if we talk about different groups, then we can talk about like selection and stuff. But let's first talk about like one group itself. So
Sure.
what is happening in this one group? So first of all, at genome level, almost all the cells, they are the same, right? I mean,
yeah, other than de novo mutation, they're the same.
Yeah.
Yeah.
So what do you think? What is the like how this differentiation is happening? So first, I mean, I think there are two questions. So first of all, like how it stops after certain time, because this is also really interesting. I mean, that's why we also, in our case, we have hands and the limbs or different
So we
organs
don't
which
have
are like.
that. Our guys always grow. They don't ever stop growing.
Uh-huh.
Yeah.
So they stop at some point, or they just continue to grow?
No, they just keep going forever until they are not passaged again. So the way to think about our population is that they're basically all under selection to grow as fast as they can. Because if you grow slower than someone else, they are going to displace you, right? They're under selection to grow as fast as they can, and they're under selection to form the biggest groups they can. And on top of that, they're under selection for groups to leave offspring groups. I'll explain the last one first. Well, actually, why are they under selection for growth rate? Each population starts out with a certain amount of food in a tube. We shake them for 24 hours. If you can eat that food faster than someone else, well, you're going to win, right? It's as simple as that. You will displace them. If you increase in frequency 1% every day, it isn't that many days until it's just you, right? If you're growing slower than them, you're gone, right? Why are they under selection for large size? Well, at the end of the day, we take out 10% of this big population, put them into a smaller tube, and we do our settling selection there. So big groups sink to the bottom first. They're the ones that survive. Not everyone that makes it to the bottom of the tube towards the end of it. Like right now, we're only giving them like 20 seconds to get to the bottom of the tube. So they don't have very long to get there, but they sink super fast. And they have to be at the bottom of the pellet. It's not good enough just to get to the bottom. They got to be the first to win the race. So being big, that's really important for winning the race. Why is reproduction important? You would think, given the description I just gave you, that the best strategy would be just to grow bigger forever and ensure that all just, survive and sink, and you could just grow forever. But that's actually not the case. We lucked into this, but this turns out to be, I think, one of the most important things we did in designing our experiment. We originally grew them in 10 mil tubes, so 10 milliliters of media, and only did settling selection with 1 mil of media. because it was a lot easier to do settling selection in a small 1.5 milliliter Eppendorf tube. And you could just do a bunch of them on the bench at once, and it was easy. But it turns out to be one of the smartest decisions that we made about our experiment. Because if we'd done settling selection in the whole tube, I think the winning strategy would be just form a giant blob, which always gets to the bottom, and all your biomass survives. But as soon as we only do a 10% of the population is competing, anything that takes that strategy is guaranteed to go extinct. A group that grows forever and doesn't reproduce has a 90% chance of not even getting to compete for settling selection every day. So what's the chance that it's around in 3 days, right? It's 0.1 to the third. It's tiny, right? 0.001. It's low. It's very, very low chance that any group will last very long in our experiments. And even though most of the biomass is in million cell plus groups, they're the lucky survivors. Most of their compatriots have gone to the sidelines, right? They're the ones that lucked out. So that's really important because it means that we have this evolutionary pincher where you have to grow fast, you have to get big, and you have to leave descendants. Because if your group doesn't leave descendants, then your whole lineage is dead. Those 3 things are really tough trade-offs. Getting big and growing fast already trade-off for biophysical reasons, nutrient diffusion limitation, etc. Getting big and reproducing is its own trade-off. What's the fine line, right? How much of your biomass should you invest in becoming a big group versus how much of your biomass should you invest in throwing off offspring? Like I said before, if these things only pursue a growth strategy, then they will guaranteed be left extinct. So each group has to essentially survive by its offspring succeeding. And so they are under selection to have a multicellular life cycle, which is really important. Otherwise, we would just get blobs and that wouldn't be interesting. What we want are things that, kind of like in nature, are pincher between important trade-offs. The lack of trade-offs gives you a lack of interestingness. Trade-offs are what makes life interesting, in my opinion, right? So grow fast, get big, have a refined multicellular life cycle. You could see how, you begin to start the cycle where you're getting bigger. And now there's opportunities for further complexity. There's opportunities for figuring out how to reduce the burden of large size in terms of growth rates. How do you make your cells grow faster in a big group? We actually think that's how circulatory systems and oxygen binding proteins and stuff evolve to deal with the fact that big groups have a lot of diffusion limitation problems. So you get these feedbacks, right? So Andy Knoll has this beautiful paper about a decade ago where he sort of argued that in the fossil record, you see these feedbacks between size and complexity. And size is a sort of driver of complexity because it creates opportunities to refine on just dumb clumps. That the cost of size is an important part of this. Secondarily, the need for a life cycle is critical because it I think also gives opportunity for division of labor. Like if you can use division of labor to regulate a life cycle, to bring that under biological control, not just have it be this random thing that, cell packing strain drives the life cycle. You can bring it under developmental control. You can begin to fine tune this very important knob for fitness.
Yeah. So, and the reproduction, I think you mentioned that it happens because of the mutation, that at certain point, the daughter cell gets a mutation and they just break off or something.
Should I show a video? That'd be good.
Yeah, you can.
Okay. All right, cool. I wasn't planning on this, but you know what? I got talks ready. I can show you a video. I think it will clarify things. Video is worth 1,000 words, right?
Mm-hmm.
So are you seeing that now?
Yeah.
Okay, great. So this is a kind of a flattened cluster. Again, you can see how it has this really nice branching morphology where this older cell has a bunch of branches connected to it. And this would be an early branching branch. And if you were to break this cell that I'm pointing with my laser pointer here, if you were to break this cell off, then it and its daughters, its granddaughters, its great-granddaughters, all of them would come floating off, right? The reproduction is actually not driven by mutation. The reproduction is driven by physics. So this is a time lapse of a single cluster growing and giving birth to babies. And basically what happens that as this thing grows, imagine again that you're building a model of a little branching tree with your hot dog, right? with ping pong balls. as it grows, you're adding cells to an already dense interior. And that's accumulating strain on those strong connections between cells. Basically, once it's already jammed, this is a specific term in physics, but once it's jammed, cells no longer have the ability to move around anymore, right? Once it's already jammed, it becomes kind of brittle. And if you add one more cell to it, then that can exceed the strength of the weakest cell-cell link in that point, and you break the whole thing in half. And once you break a single cell-cell connection, and just like sawing a branch off a tree, if you break one cell-cell connection, then you basically take that whole branch, and where the break occurs, it and everything downstream just falls off. So you get this branching growth form, which has a built-in life cycle, which is emergent. It didn't evolve for a reason. It's just a consequence of physics. Cells divide in 3 dimensions. Cells grow as branches. Eventually, they get overly densely packed and they break. And because of the topology, specifically topology, I mean the mapping of how these cells are laid out as a network, right? That wherever you break a cell-cell connection, that cell that's the most basal cell in the branch, the one that flew away as the propagule, the the oldest cell there, where it broke away from its mother cell, that is the parent of everything in its group. For those that are watching this in the video, you can kind of see in this group here, if I was to break, pick any cell in your mind, right? If that cell and its offspring were to float away, you would see how, oh yeah, like that whole branch that floats away has a single cell ancestor, it's a single clone ancestor, right? And as a result, if this group starts out chimeric because there was a mutation somewhere, and there's a whole branch now that's mutant, every time it divides, there will be a group that's generated that's only clonal. If the breakage occurs between 2 wild type cells, well, then you have a wild type only propagule. If the breakage occurs between 2 mutant cells, you have a mutant only propagule. And if the breakage occurs between the first wild type and the first mutant cell, now you've cleanly separated out these mutations. But at most, when you have a new mutation arise, it can only ever be chimeric. That is, the group will contain both the wild type and the new mutant in one group. So if I were to take a single chimeric cluster and seed it into a population and watch it grow, I could have 1 million groups grow as a result of that first parent, and all of them but one would be clonal, because it can only make clonal offspring. Again, importantly, that is not evolved for a reason, but bottlenecks are incredibly important in the transition to multicellularity because it allows group level traits. So it does 3 things. Number 1, it allows group level selection to be efficacious. If the group was a random grab of the cells, if it was representative, if the cells in a group were representative of the cells in the population, then selecting on the group doesn't really do anything. You're not changing anything by selecting on a group, right? If there's 50-50 ratio of A and B cells in a population, and every group has a 50-50 ratio of A and B, groups don't matter. Doesn't matter. You can select on them all day long. It doesn't do a single thing, right? If groups are only A or only B, and all A's are favored and all B's are left behind, well, then group selection completely purged the B genotype, right? The more clonal your groups are, the more group selection matters in terms of evolution. Number 2, it allows for emergent multicellular traits to have a genetic basis, which allows emergent multicellular traits to be heritable. If every cell in a group shares the same genotype, and now you have a mutation come along in this population, which changes the way a cell behaves. Let's go with the example I'm using here. It makes the cell more elongate. More elongate cells, the change in the cell shape changes the way that cells pack in 3-dimensional space. Change multicellular packing changes the size to which the groups grow before they break. Bigger groups, all of a sudden, hey, they have an advantage in our environment. Bigger groups have a higher survival. Selection favors large size. In this case, what makes large size heritable is the fact that our groups are clonal. Every cell in that group has the same genotype, but these emergent multicellular traits, right, are what selection is seeing and acting upon, but it's correspondingly acting on the genetic basis because of clonality. And number 3, this is less an issue for us, but it can be an issue in other systems, is it prevents genetic conflict. You don't really have the possibility for cheating or, sort of cancer-like dynamics where some cells in the group are doing something which increases their short-term reproductive gain at a cost to the lineage or the group as a whole, if every cell is a clone mate. A group of all cooperators does great. A group of all cheats does terrible. A group that's mixed has problems, right? And so again, we don't really have cooperating and cheating as an issue in our system. I think you could imagine a scenario where it would be could be a problem, but clonality just nips that in the bud. You just don't even have to worry about it at all because it doesn't have a chance to get going.
Yeah, but this definitely highlights the importance of cooperation or symbiosis in the system, right? Because we can think of systems where certain cells can be parasitic or something with the other cells, and whether those strategies would evolve in like something like multicellular system. I mean, we can think, especially we can take the example of evolutionary game theory. I think that's like the best way to think of it. And again, we will reach to kind of this conclusion that it'll be like moderate, like this 60-70% cooperation. And so there is this one solution for in human altruism, where they consider that if it is like 60-40, this is like the best strategy in a society. So I think that like
Probably depends on the specifics a lot, in terms of what the sort of Nash equilibrium will be, and then what you want you want in terms of the system. I think in some cases you want only cooperation, right? Like cancer would be a good example of like, I'd prefer 0% defectors, please. Thank you.
Yeah. You're right.
But
it is system dependent.
Yeah, it's going to be. I mean, it's system. I think that's true for cooperation writ large, or biology writ large, right? So just to sort of like touch this back, like one of the things that I like about our system is that we sort of went into it not trying to force a specific outcome. You know what I mean? Like if I was doing only sort of synthetic biology, I could create something which I already kind of knew would work and then show that it works in biology, which is great and a nice way to test hypotheses. But, we didn't do that. We kind of said like, okay, I have a cell. What would happen if I challenged it in this environment for fast growth and group formation, large group formation, and having a life cycle? How's it going to deal with that? And, they basically adapt really well to it. And not only do they adapt well to it, they sort of just walked into an almost optimal solution. This multicellular life cycle that they have that Snowflake yeast have is really, really good at being capable of having open-ended Darwinian evolution in response to these challenges. And it's certainly we're not trying to explain all different transitions to multicellularity. Multicellular world is way too diverse for that, right? We have bacteria, we have archaea, we have eukaryotes that behave like slime molds, we have eukaryotes that behave like plants, we have a lot of diversity. We're not trying to sort of explain it all. But a lot of things started out with small branch structures, the way that Snowflake yeast do. If you look at directly through the fossil record or infer through phylogenetics, the earliest forms of multicellularity and the lineages which kind of ran away with complexity, plants, not animals. We don't know what they really looked like in the early stages. It's a big gap in our knowledge, honestly. But plants, fungi, the seaweeds, they looked a lot like Snowflake yeast early on. They formed essentially branch structures. And so I think a lot of the sort of benefits of this life cycle that we've seen with Snowflake yeast very plausibly could have gotten going and provided that sort of Darwinian closed loop in these lineages that ultimately evolved to things that look really, really different.
Yeah. Are you familiar with Michael Levin's work? Because
in fact, we're collaborating.
Oh, interesting. So because this is what he did, or his collaborator, that they could form Xenobots where they could just take the skin cells of frogs and like it forms this clumps which actually behaves like Xenobots that they So
totally.
It sounded like the similar kind of system going on in the case of Snowflake yeasts, and the only, or I don't think so that there is much difference there in terms of underlying principles, how the cells they come together and stuff.
They're
Apart,
pretty different in terms of they're similar in some ways, they're very different in other ways. So like their behavior comes about more because they knew it ought to work, right? So they have this computer aided design where they basically simulated different morphologies that ought to have different behaviors, and then they created those morphologies, like surgically, basically, and confirmed that they had the expected behaviors that you would get from the computer. I think the biggest difference is that their system is not really a model of multicellularity in the same way, and that it's not Darwinian. So their groups basically live until they die, but they don't reproduce. And the variation that they have in their multicellular traits, which depends on their shape, is completely non-heritable. It's created by the experimentalists. That's where the variation comes from, but there's no genetic basis for it. And so they don't reproduce and they don't have heredity. So they're truly a non-Darwinian system, which is really different from ours. Like ours is like 100% Darwinian origin, right? And theirs is 100% non-Darwinian origin. That being said, what I think is really cool, well, one of the things I think is really cool about this work is how it sort of shows the like weirdly latent potentials in living systems for like novel forms of function. Like if you can take skin cells that have existed in an animal for hundreds of millions of years, ball them up, and all of a sudden they can like start doing completely different behaviors. It just sort of shows, like, really highlights how plastic life is, and how living systems are able to kind of like, it's kind of gets back to this emergence thing, right? Like they're sort of they're emergent traits actually that they knew should work, right? So they designed it this way, but the behavior of their group is kind of an emergent phenomena of how cells behave, right? The cell level. And it really does highlight just how this is why life is so persistent and so good at filling different ecological niches. It's really plastic. It can take really weird turns. I might have predicted that animal cells would be much more stuck on doing what they've done in developmental terms for a really long time, and just wouldn't be as plastic.
yeah, I mean, there is definitely a lot of plasticity, and especially after reading about Michael Levin's work, that was like the interesting part. That okay, that some of the cells they can come together and form something like these bots. But I
Yeah.
mean, of course, this is again another interesting point about multicellularity that you mentioned, that the Darwinism is important. Like if you want to have an entity there, Darwinian evolution should kind of work on it or work on that system.
Yeah, it's
Right. Like this is,
it's the framework through which we understand all multicellular lineages to have arisen, and it's I think everyone agrees that it's a necessity for getting multicellular life. And I think the question really is like how do these things occur, right? Like there's another sort of framework for how early life cycles arise that's very different from the one that I've described to you. It's Paul Rainey, Andrew Black, Pierre Borat's ecological scaffolding framework. And so here they basically argue that, and I think there's merit to this, that basically environments can create the life cycle initially, and that later gets endogenized by biology. So there's a recent paper in eLife, which I actually, me and a grad student wrote like a little primer on. I think it's a really cool paper, which basically reports on the evolution of a new kind of multicellular bacterium. It lives in a Japanese cave that gets periodically flooded. They isolate it off the wall of this cave. When you grow this bacterium, it sort of forms this iridescent sheet of cells, and all the cells are lined up like a little liquid crystal. Then in the middle of the sheet, it actually starts to bulb up, and it's producing a whole bunch of circular cells. The other cells are really long and skinny. Bunch of circular cells. When you submerge this thing, they start to shoot out these circular cells from the biofilm the way that like puffballs shoot out their spores, but only upon submergence of water. And presumably these things would then be dispersed in the currents of the water. They would wash up on the walls of the cave, where they would then regrow and form these new biofilms that would differentiate and have, the life cycle all over again. So here you could imagine that this is like a really nice environment where the periodic flooding creates the life cycle. Imagine way before we have this organism, you just have something which forms biofilms, and every couple weeks it floods, and the cells are dispersed off. They settle back onto these walls of the cave. Every cell is now far away from any other cells, and as it grows, it forms a clonal group. Bottlenecks, right? We have a clonal group of cells. Perfect. Now, if those groups vary, like you have mutations that varies in their group level properties. When it floods, selection acts on those group level properties. You could see how we have kind of a life cycle, sort of like the one that we have in Snowflake yeast, but it's created by the environment. Then later it gets endogenized by mutations, which basically co-opt the life cycle and make it better, refine it. So I think it's a really appealing hypothesis for one way that you could get life cycles. And until now, I don't think that there was any living system where you could kind of point to it and say, like, this is how the ecology scaffolded its life cycle. This is how the ecology actually, like, we couldn't point to that for plants or animals or Volvocene green algae or fungi. Maybe for things that have aggregative life cycles where switches between starvation and non-starvation play a role, but even then, I'm not totally sure. This cave study is fantastic. Mizuno et al. I would look it up. It's a really cool paper. It sort of illustrates how scaffolding would work. So I think, I don't know if that actually played a role in any extent lineages. I don't know if the Snowflake approach, which would be like a biophysical life cycle, plays a role. But certainly the biophysical approach I think has the advantage of being like really strong. Like, it has the advantage of, like, there's no variation. Like, if you're in this group, cool. All of your offspring are in groups too. They're all in basically clonal groups. They all reproduce at the same size. Like, it's really a powerful life cycle. Whereas the scaffolding, the devil's really in the details, right? Like, how much variation is being mixed. Like, if you have these cells not well spread apart, but like a bunch of cells are put into the same spot, well, then the bottlenecks are really weak, and it's probably not going to work so well. Like, you need really nice separation between groups. How often does that actually occur? What's the interval? Are you really dependent on the environment having a nice regular periodicity? The devil's in the details. The biophysics stuff is nice because it's innate and it's very powerful.
But the properties of the multicellularity, I think that'll be kind of the same. Thinking in terms of even in terms of snowflakes, for example, the individuals which are like kind of at the center, they would I don't know. So what will happen in their case about their reproduction, for example? How do
Yep.
they get nutrients?
Yeah.
Even access to environment in general. So.
Yeah.
What's your thoughts on it?
So yeah, I got a lot of thoughts on this. Some of it I have to be a little bit less forthcoming than I'd like to, because I have some projects going with collaborators, and I'm not sure what their feelings are on sharing some of this data,
Uh-huh.
But let's just say this: our evolved snowflakes, the ones that are big, like, the size of Tic Tacs, like, they're millions of cells, they're huge, they're way bigger than they should be able to be based on diffusion limitation alone, seem to have circumvented diffusion limitation to a large degree by figuring out how to actively flow water through their tissues. They look like sponges. They're shooting water through themselves. And we think we know the mechanism, but I don't want to say more because I haven't cleared that with my collaborators. But so I think they're figuring out some hydrodynamic solutions to the problem of diffusion. Hopefully, actually come to the APS March meeting. We'll tell you there.
Mhm.
So that's okay. So then, so one of the types of divisional labor that like, remember I mentioned that we have entanglement. These things are getting really tough, right? Because they're wrapping cells around each other, which actually breaks the biophysical mechanism of the life cycle. The life cycle is grow until fracture breaks a branch, right? But if they entangle, fracture doesn't break branches anymore. That's great for getting big, but it's bad for having a life cycle. Groups just keep growing, but they don't throw off offspring. Okay, so that lineage now has zero fitness. If you don't throw off offspring, you have zero fitness. So how do you solve that? Well, it turns out that about 1,000 1,000 generations after they evolve entanglement, we see like really high rates of programmed cell death arising. And the mechanism they're using there is really interesting. These guys are growing under anaerobic conditions, and they got rid of their mitochondria. Their mitochondria, they still exist, but they're teeny, and their mitochondrial genome is shed. It's basically, they don't need it anymore. It's gone. But a lot of the mitochondrial proteins are what causes programmed cell death, and they're actually stored in the host genome. And so these guys are cranking up expression of host mitochondrial proteins, which causes high rates of programmed cell death. Programmed cell death causes breaking of branches. And what we're seeing is that our guys that are big are shedding thousands of like 5-celled propagules. Rather than kind of growing and breaking in half, they're just shedding tons of little tiny guys. We don't yet know if this is an adaptation or if it's just, a side effect of something weird. But it's consistent. We see it evolving in different lines, and we see it being maintained over evolutionary time, which suggests to me that it's not a fluke. And it would be interesting if this is their solution to basically, they started out with the life cycle where they grow and they break a single cell-cell connection, and now you have two. That's how they reproduce. Then they evolve entanglement, and that's no longer the case. You no longer have the biophysical life cycle, but you need a life cycle, because if you don't have a life cycle, your lineage has zero fitness. What do? Well, they may reinvent it by using programmed cell death as a mechanism of actively shearing, essentially breaking lots of bonds, but that mostly essentially breaks the only things that fly off are the unentangled surface, because the surface cells are not yet entangled. Only the interior cells are. And if that's true, if basically the only cells that are surviving are those that are the only groups that are really making it to the future are those that are little baby propagules in the surface. And I don't know that that's true yet. This is work in progress. Then it actually sort of suggests that there's a big shift in the way that our snowflake yeast are evolving. Like initially, being in a group was good for every cell in the group. Being in a group meant that the group grows, it splits in half. The cells that are in that group have a higher chance of surviving into the future because, hey, the group is like a vehicle for getting cells to the bottom of the tube, right? It improves their long-term individual survival in groups. But once you have an entangled group, by the way, I don't know if this is good or if logic is great or bad yet. I'm really working through this in my own brain right now. Once you have an entangled group, the cells in the interior, they may never, ever have the chance to escape that group again. They're stuck. They're entangled. I don't think these entangled groups are ripping themselves in half. So I think the cells on the inside are there until the group dies. And all of their future reproduction comes, like their future fitness comes not from themselves essentially getting into the next group, ripping the group in half and being in the future. It comes from their offspring that are like their clonal offspring, but like 3-celled propagules that are being shed. That's where their reproduction comes from. Not from them surviving, but from their great granddaughter cells being the ones to make a little baby. If that's true, it's really interesting because it actually reflects a very consistent change that we see in natural populations, where we go from early multicellular groups, where basically group formation is a way to increase the survival of individual cells. If you're in a group that doesn't get eaten by a predator, great, you survived, right? To one where cellular differentiation, morphogenesis means that the cells in groups are there to support reproduction, but they themselves aren't passage from generation to generation. If you think about humans, right? You got 1 trillion cells, just dump them all. Like only reproduce through one. Just forget about the rest, right? Just reproduce through one. And plants do the same thing. Fungi do the same thing, right? I mean, a lot of them reproduce asexually, but a lot of them make a sporangium and shed a whole bunch of single spores, and then the mushroom fruiting body just dies, right? This is really, it's a weird thing to think about. Why would you invest 1 trillion cells in a body and you throw all the cells away just to preserve 1 or 2, a small number that become the next generation's propagules? That's a huge derived trait. It doesn't make sense from the logic of early multicellularity, but it does from the logic of having complex bodies. And we may be sort of on the road to that, which would be ridiculously cool. Like if we're evolving soma just because that's what's adaptive, that'd be cool. What are the consequences of that? How does that affect the evolution of aging? Blah, blah, blah. I mean, like, I think there's a million questions one could ask. So it's still relatively early days for the experiment, where we are 1,400 transfers in total. We've rigorously analyzed up to about 1,000. We have papers in review up to 600, from 600 to up to 600, from 0 to 1,000, analyzing different aspects of it. It takes years to really actually do the analyses and write the papers and respond to reviewers and all that. So we'll always be, behind the leading edge of where the experiment is. But yeah, it's still relatively early days. We're only 5,000 generations into this experiment, and it'll be very interesting to see what happens by 25.
Yeah, only 5,000 generations. Yeah.
Only 5,000. You too can evolve microbes through thousands of generations.
Yeah. So what about the polarity part? Because I think that is again another important aspect in the case of multicellularity and differentiation, especially I think I was checking Nicole King's work, and Nicole King's group at University of California, that they are working on Choanoflagellates, and I think she talks about kind of this polarity emerging with the Choanoflagellates forming this multicellular
I'm not familiar
entities.
with the polarity argument for Choanos. I'll have to go look through her stuff again. I actually co-taught a course with her this summer at Woods Hole, and it was a wonderful experience. We did like a 2-week lab rotation where we took students into a joint group and had them do experiments on Snowflake, yeast, and Choanos. Yeah, it was great. I'm not so familiar with polarity in Choanos. I wouldn't be surprised if there were. There is definitely polarity in other holozoan simple multicellular groups. So Omaya Duden has shown polarity in a ichthyosporian. I think that work's unpublished as well, so I shouldn't say more, but looks like an embryo. It's really cool. I think for polarity, in our case, there's really no incentive to evolve polarity. Like, they're in a well-mixed, shaken environment where they're being tumbled at, 1 centimeter per second, right? They're flying all around through space, and it's incredibly chaotic. So for them, we don't see any polarity, and I doubt that we would really get it, because if we got it, I wouldn't be surprised if it makes sense, hey, post hoc, sure, wow, that's how it works. But I wouldn't necessarily expect it, because they're basically growing in 3-dimensional space, kind of as spheres. Maybe there's an advantage to having like a football shape during the hydrodynamics of settling or something. But they're not really under very strong selection for polarity. Then it's not like they're growing towards a resource that's over there, and they have to figure out how to like only grow in that one direction or something, which would be a pretty common, or swimming towards a mate, or swimming towards the light, or forming a specific body plan that requires polarity. That's not quite the kind of multicellular environment or organism that we've put them under.
Yeah, but once we start thinking of this like polarity part, because definitely we have it in many life forms. So what is the bottlenecks of that? It's whether it's like genomic bottleneck, or it's like the physical bottleneck, which defines kind of, or it's basically both physico-chemical bottlenecks. And when we think about this question, I think the answer will come from system like where we are trying to evolve multicellularity, right? Because it'll be really cool to understand what are those underlying principles which explain this kind of polarity part.
Yeah, I agree. It would be really coal. And Omaya is planning on doing experiments with something which already forms polarized small groups of cells that look like embryos. And I think he could learn a lot from those experiments, or we all could. My guess is that the mechanisms are going to be very system dependent. Like, they're going to depend on the cell biology available to the ancestor, the environment, and you wouldn't necessarily get the same thing if you started out with an algae, or animal relative, or a fungal relative, or a bacterium for that matter. And that's kind of true multicellularity. Like, the genetic details really depend on the ancestor, like on what the ancestor brings to the table in terms of toolkits, right? That's not just multicellularity, that's life. That's evolutionary biology, across the entire tapestry. LUCA matters because it provided the basic toolkits for everything. The same genetic code, same Krebs cycle, same
Yeah.
plasma membranes. um, so yeah, so those contingencies are extraordinarily important. Yeah, unfortunately, I don't think Snowflake yeast will answer that question. If
So
they
since
do figure out some kind of polarized growth, though, that would be amazing. But I'm not expecting.
I mean, of course, both prokaryotes and eukaryotes, they can form multicellular organisms, if we call them organism, or at least things, as we decided to call them. So one interesting aspect is this syncytia, like
Yeah.
multinuclear cells, because I think that's the important aspect. So what is happening there? First of all, are there any system studies where they see multicellular systems with the multinuclear cells? And I mean, of course, then there are again a lot of topics there to cover, but at least at the multicellular level, what do we know about multinuclear cells?
I think there is stuff known about this. So okay, I'm gonna have to do a little Wikipedia search for this thing. I forget the name of it. It's very frustrating. One of them is the whole class of cellular slime molds. So like Physarum polycephalum is the dog vomit fungus that people love to. You'll see it in your mulch. Like, what is that giant orange blob? You come back from lunch, oh, it's gone. So there are these slime molds that are called acellular slime molds because rather than unlike the cellular ones, which actually are multicellular and have a bunch of cells that are discrete, you can have these like basically have what's kind of multicellularity via a different route. The formation of a gigantic quote unquote single cell because it's one bounded blob, but it's the size of a volleyball. And there's millions of nuclei and it's modal and basically crawling around decaying organic matter, excreting enzymes and shredding bacteria and fungi and eating them. They're really cool. And it's a syncytium. It's not multicellular, but it's related. It's basically taking advantage of similar behaviors. It's very animal. I mean, the acellular slime molds are very animal-like. All right, I gotta figure out how to find this. Okay, what is this called? So there is.
Are
Oh
you
yeah,
looking for us?
Caulerpa. You know Caulerpa?
Caulerpa, yeah.
Caulerpa is a great example. So
Yeah.
if you saw Caulerpa, you'd think, oh yeah, cool, that's a seaweed. It's got a stipe like a rammet. Basically, it's got like a underground a root that grows along the surface of a shallow marine environment. It's got big fronds that come up off it. It's in fact an invasive species in some parts of the Mediterranean, I believe. And it's one giant cell. Like it looks like a multicellular plant algae, but it is just one gigantic cell. It can be meter length, meter scale, one cell, which is really cool. I think without that example, I would have probably said, yeah, without multicellularity, sure, you can have dog vomit fungus, but you're not going to have something which is like, has sophisticated morphogenesis at meter scales. But hey, here you go. Life figured out a way to do it with a gigantic single cell. It's wonderful. I actually wish more research was done on that organism. I think it's really, really cool. But also it has, one of the things that's completely consistent, and I don't think there's any way of getting around this, is there are giant bacteria, single cell bacteria that are centimeter scale. There are meter scale Caulerpa, single cells, but they look like a brown alga. There's the dog vomit fungus. In every single one, they're highly multinucleate. The ratio of nuclei to cytoplasm is roughly conserved across all life, And I think the reason for this is that you basically need to coordinate the functions of these cells. And you can't really rely on sending mRNA. If you have a meter length cell, you can't rely on sending mRNA from a single central nucleus all the way out to the tip. It's too slow. How would you coordinate? You can't rely on diffusion over length scales like that. It would take forever. The cell is a densely packed place. So they basically are kind of behaving like multicellular organisms, but they don't have the cell boundaries between these different sections. We still have local behavior. You still have many nuclei. So yeah, we can't call them multicellular because there's only one cell, but in some important ways, they share common characteristics.
So, so at the end, once we want to understand like what is the importance of multicellularity and how does it evolve and all that, right? Like these are the basic questions. So right now you are using Snowflake yeasts as a framework, but are you planning to, I don't know, use other systems? Or according to you, what are like the major questions which can really nail the problem and, which can give us like these underlying laws that can help us to kind of generalize it to quite an extent? Of course, there is a lot of plasticity and we might not be able to explain everything, but at least to quite an extent what we can explain.
Yeah, it's a good question. I think I'm going to stick with Snowflake yeast primarily. There's real benefit in specializing on one system, and it takes years.
Yeah.
It takes years to work the kinks out. A bunch of something weird happens. I love lab mysteries where someone's like, "I did this experiment, I got this result." I'm like, "Great, that makes sense." And someone else is like, "But I did this experiment to test the hypothesis that we all thought we didn't need to test, and it's not the way you think it is." Like, wait, what? It turns out 2 years later, like you learn something really exciting but completely unintuitive. And that's really common for me with Snowflake yeast. So yeah, I'm gonna keep working on it for the rest of my career. As long as I can secure funding and have students that want to work on it, which hopefully is possible. And I think it will continue to bear really exciting fruit. Like the longer it's evolved, the more we have this frozen fossil record. We can go back at any time point in the experiment and pull living cells out. So you can, in principle, interrogate any question you have about it, even things that we can't imagine we would want to ask right now, which is really powerful. And I saw in Rich Lenski's career, that was huge. He started his experiment before DNA sequencing was a thing, especially high throughput. Whole genomes? No, you weren't doing that in the 1980s. That wasn't happening, right? But once that was an option, it just cracked open like so many new exciting things that they could do with the resource of investing decades in an experiment like this. I'd like this to be a similar resource, something which it's not just our lab working on, but we can also collaborate with others. We can provide samples to others that want to ask questions that we're not working on. I think that'd be really cool. Like I'm not someone who feels like if I didn't do it, it doesn't matter. I think that's really costly to science. So I would much rather this be a resource for the field of evolutionary biology, and that other people were excited to work on it too. That would be the biggest win, in my opinion. Okay, so big picture, but I want others to start their own experiments. Like I would love for other young scientists to be like, great, I'm gonna Amaya Duden, I'm gonna work on this crazy holozoan and I'm gonna make it an evolutionary system, or someone from Nicole King's lab doing Choanoflagellates, or maybe Nicole herself as well, although she's not doing experimental evolution, but maybe someone would want to take that up. So we've actually done some work on algae. We worked on some green algae that are related to the Volvocene green algae, which are a clade of multicellular algae. And I collaborated with Matt Heron, who's a Volvocene green algal scientist on that. And it was cool. I think we learned some nice stuff, but I didn't like working with algae. I really like yeast. So I'm happy with my niche being Snowflake yeast. Big questions though, I think, we have the framework. We have an evolutionary framework. The framework is you start out with a single cell. You get group formation, likely through genetic means. Otherwise, it's probably not very persistent. You get mutation, which forms groups. You have a simple group. Groups become Darwinian multicellular individuals. You go from a multicellular group to a multicellular individual. How does that occur? How do those life cycles arise? How do the traits of those life cycles affect the potential for evolution in that system? I haven't talked about this at all, but we have a paper submitted last week to eLife in which we basically engineered a clonally developing snowflake and an aggregatively developing flock yeast. And then we evolved each one in parallel. We basically showed that the snowflakes mostly underwent group adaptation, and the flocks, which form chimera groups, mostly underwent cell adaptation. So, you can get groups and they can be Darwinian, but the way in which the life cycle forms can really affect downstream dynamics, right? So understanding the role of early life cycles, how they form, how they constrain or catalyze future dynamics, I think that's really cool, really important. I think the biophysics of increased size is cool and underappreciated. Not a lot of people are asking, how do simple multicellular organisms become tough? Or even honestly, how extant ones do. Entanglement, is something which has barely been studied, even in extant organisms. And I see it all over the place, but no one's done the experiments to sort of confirm that they really are entangled. I think maybe one of the most important, if not the most important question, is the emergence of development and cell type specialization. Like that's kind of what makes multicellular organisms so powerful, right? That's why they're so cool. They were all one cell type, we wouldn't be that jazzed about them. Be a big blob. But the fact that there's multiple cell types and there's specialization, that opens the door for biological complexity in a way that, well, outside of Calerpa, you don't really see in the single cell world. Calerpa, you're awesome. You're messing up my pitch here, but you're awesome. But also genetic and bioelectric, back to, stuff Levin's worked on, bioelectric mechanisms. I think those are both really interesting. One thing we haven't talked about, but is really important, is understanding entrenchment. So how do cells get stuck in a multicellular state? Like, why are there no unicellular descendants of animals or plants that we know of? They've been around for a long time. Animals have been one of the dominant clades on this planet for 600 million years. And there are no single cell descendants that we know of. It's not like there aren't, a vast amount of cells like in the environment, but they don't seem to revert. Why is that? Is it ecologically they can't compete? Is it that they get so stuck on living in a group that they simply die if they're separated? I mean, plants are a little more plastic there, right? A lot of plants, you can cut a branch off, stick it in the ground, and it grows new roots. And yet you still don't have single cell descendants of plants, right? So that we call that entrenchment, and understanding how entrenchment arises and what its consequences are, I think is really cool, both because we see it evolving repeatedly, and it lends directionality to this transition, which I think that's underappreciated. The fact that there seems to be directionality with major transitions. Sure, there are multicellular organisms that evolve back to single cells. Fungi do it all the time. They love to go back. But generally speaking, there tends to be directionality. And I suspect that it has to do with division of labor and differentiation. No one's really tested that or even done a phylogenetic analysis of that. How do different environmental drivers affect the evolution of multicellularity? I think that, what are the constraints? So we know that cells evolved on Earth at least 3.5 billion years ago. My bet would be 4 billion or earlier. Like, we don't have the evidence, but like my money's on pre 4 billion. And the multicellular biosphere pre 1 billion years ago is pretty lame. Yeah, there's like some little things going on, but not much. Filamentous cyanobacteria, filamentous algae, just nothing big, nothing sophisticated. And the last billion years, eukaryotes go crazy. Is it eukaryotes? Is it oxygen? Is it something else? Right? We really don't know the answers to those questions. Those are important. How does, anyway, so that's something we work on too. We work on the role of oxygen and light, the evolution of multicellularity. And then I think one that, we've focused almost exclusively at the organismal level: multicellularity, multicellular individuality, multicellular organisms. We haven't talked about multicellular ecosystems at all. And yet, I think you could make the argument that just as important, or maybe more important than the evolution of complex organisms themselves, are the ecosystems of multicellular organisms that multicellularity allowed to evolve. Most of the carbon on the planet is locked up in plants. Plants basically did super well because they were the only algae that could survive to live outside of water. Not the only ones, but they're like their innovation was not living on land; it's living in the air. And once they lived on the air, they have this huge, vast frontier. Plus, you get all this ecology, diversification, adaptive radiations, right? the building of these new ecosystems, which we call by the dominant multicellular organism that lives there: forests, coral reefs, right? Grasslands. We use the shorthand of the dominant multicellular organism to define ecotypes, ecosystems, right? Like, that's cool. What part of the transition to multicellularity sort of allows for the creation of whole new ecosystems around multicellular organisms? Is it just that you have, is it like plants where they're just opening new terrain, or is it the fact that you have more opportunities for life histories and lifestyles once you have the ability to be multicellular, or combination of the above? I really don't know. Maybe that's more of just a fundamental question for ecology, not taking the evolution of multicellularity for granted. Like, a lot of these questions are worked on by ecologists, but from the perspective of like, once we have these, what happens? But like, that was game changing for planet Earth, the origin of multicellularity in terms of the ecology, not just the organisms themselves. So I think those are all outstanding questions for the field, and I would like to try to address as many of them as possible in the remaining time that I have. But I would also love others to join us on the journey.
Yeah, I mean, definitely there are a million questions when we think of this topic, because I mean, certainly the topic connects both, kind of origin of life and all that stuff, starting from the protocells, simple form of life, to all the complexity and diversity that we see around, and the kind of this emergent complex behaviors that we get out of the systems. That's just fascinating.
Totally.
And I mean, it was supposed to be my last question, that whether we can reverse the process.
Yeah.
Whether we can, but now you've already covered it a bit. But what do you see in your like Snowflake system? Do you see at some point these multicellular, some of the cells that they kind of go back to unicellular?
So if we try to force them to go back, it gets harder and harder. So this is something that Peter Conlon, a postdoc in my lab, is working on. The longer they've been multicellular, the harder it is to make them revert, and the lower the fitness of the revertants when we do get them. So we think we both have like mechanisms that prevent reversion because they just have more redundancy mechanisms for forming groups. But also, the cells that are really fit in groups are not the same. They don't have the same traits that cells that are really fit as growing as single cells are. We have these things that look like hot dogs, not spheres, and their cell cycle's all crazy messed up, and that's how you get elongated cells. And they got to push in order to divide, they have to push nuclei with a mitotic spindle like 2.5 times as far. And when we measure the time it takes like an extra 30 minutes to just even divide your nuclei, right? Like, but you need that if you're going to make a big group. And so certainly group adaptations seem to carry cell level costs, that if you were to compete them at the single cell level, they might be ecologically at a disadvantage. It'll be interesting to see if we get to a point where we have syncytial connections, and so breaking them apart just causes them to like die. It seems very possible that they'll become more sort of filamentous fungal like, and therefore trying to make them single cell just means that they bleed out of both ends.
So basically fusing the membrane and
yeah, they're not doing that yet. To be clear, we don't see them doing that even at 1,000 transfers. But I wouldn't be surprised if they did essentially evolve to have open connections between cells. And then if you were to somehow get them to either chop them up as single cells or genetically force them to divide as single cells, that they would be all kinds of messed up, which would be more akin to what happens if you tried to scrape your own kidney cells off and see what happens. They wouldn't do so well.
And of course there are still many questions, but thank you so much for accepting the invitation and
Thanks for having me
sharing
on.
all the exciting research and the data that you have. It's
Thank you. I'm delighted to be here. I'm honored to be here, and good luck with your studies too. I hope we get a chance to meet in real life sometime.
Yeah, sure. That'll be great.
Cool.
Yeah.
All right. Thanks.
Thank you.